Hello Hires, this is Unit 1 Subtopic 2, Replication of DNA. Okay, so first to get you started, have a think about why do we need cells to divide and what must happen before a cell can divide. There's a little fact there for you. It is estimated that an average adult human is made of 37.2 trillion cells. And there's a little picture there of one cell, obviously very simplified, turning into two. So why do we need cells to divide? Hopefully if we were in the class, you'd all be sticking your hands up and saying, well, if we want to grow, if we want to repair, we need new cells for that. More importantly, for this topic is what must happen before a cell can divide. Right, so that's kind of the starting point for this lesson. Right, so this topic is all about how DNA manages to make a copy of itself. It's called replication of DNA. And after this very long video, you should be able to state the requirements for DNA replication, describe DNA replication of the leading strand, that'll mean nothing to you just now, and the lagging strand, similarly, you won't have a clue what I'm talking about just now. State some uses of PCR and describe the process of PCR. So there is lots to learn in this lesson. Right, so back to sort of our starter questions. This is Nat 5 revision. Before a cell can divide, the nucleus must divide. And before the nucleus can divide, the chromosomes must make a copy of themselves. So hopefully you can remember some of this from National 5. So if we said this is the parent cell or the original cell, and there's 4 chromosomes in there, although you can't really see them, how many chromosomes would there be in each daughter cell? And you're all shouting at the video, 4. If there's 4 chromosomes in the original cell, then each of the new daughter cells also has to have 4 chromosomes. You know that from National 5. Oops. Okay, so you could also tell me that chromosomes are made of, shout it out and I'll click the button, DNA. So when we're going from this stage here, stage 1 to stage 2, my arrow's twirled and gone wonky, then we know that the chromosomes have doubled, and if chromosomes are made of DNA, then what's really happened is the DNA has replicated. The DNA has made a copy of itself. Because remember, the chromosomes are made of DNA. So if we had 4 chromosomes here, and we've now really got what's going to become 8 chromosomes, because we need to have 4 to go into each daughter cell, then the DNA must have made a copy of itself. Now remember, this isn't important for higher, but remember when the chromosome does make a copy of itself, each copy is called a chromatid. So we would still say that's 4 chromosomes, but each one is made of 2 chromatids. Often, if you look up pictures of chromosomes, you see them drawn like that. They sort of look like funny little elongated X's. That's just them showing them after the chromosome has made a copy of itself. The DNA has replicated there, so just so you don't get confused between different pictures. Right, so this diagram shows DNA replication in action. Right, now I've not told you anything about it, so I just want you to look at it and try and work out what's going on. We know that DNA exists normally as a double helix, so that still looks like a double helix at the top, but at the bottom here, something's happened. And then the green arrows are showing something else happening. Okay, now if I show you this, so that's the same diagram you just looked at. This is exactly the same thing, just happening that the person that's illustrated it has just made it go horizontally rather than vertically. It's the same thing that's going on. So don't worry about what direction they're sort of drawing the picture. You need to know your facts correctly, and if you do, then you'll not go wrong. Okay, so how does DNA replicate? Now, this diagram here is very simplified. So here we've got one strand of DNA, and you can see it looks like it's starting to separate out. and it looks like there's new bits of DNA, that's the bit in red, magically appearing on each strand that's separated out. Okay, so this is very, very simplified, but if you just start to think about what's happening, the DNA strand, the double helix, is unwinding and it's becoming separated. And new strands are growing. And what that means is, from our one original strand here, we're going to end up with 2 strands. And each new strand is going to have one of the original strands in it, the blue one, and one new strand, which is the red one. Okay. Now, this makes it look that like it just sort of starts at one end, finishes at the other end, and it's very simple. It's not quite as simple as that. In reality, this process is happening at several locations along a linear chromosome. Remember, linear chromosomes are the ones that we find in eukaryotic cells, and that's the only type we're going to be worrying about now. This is just another diagram to show you that. So here we've got DNA in our double helix at this side, DNA in our double helix at that side, and that would have, if we could rewind time, that would have all just been double helix as well. But this section is in the process of being replicated. So what's happened is the strands have separated out and something's going on in here. This is just the exact same diagram, but it's just shown 3 of those. So this is our big long strand of DNA, and in these sections, replication's happening, and it will continue to happen. Right, so let's think, what do we need for DNA replication? What are the requirements for DNA replication? So if we think about it's going on inside a cell, so what do we know controls everything in the cell? What do we know sort of is in control of reactions in cells? What will provide the energy? Hopefully you're thinking of something there. And what are the obvious things? If you want to make a copy of DNA, what are the obvious things that you're going to need, thinking about the structure of DNA and what it's made of? So the requirements for DNA then are enzymes, because we know that they pretty much allow all the reactions to happen at a suitable speed. ATP, because that provides the energy within cells. We need the DNA template, that's the original DNA. We need free nucleotides, because they're the building blocks. We can't make any more DNA if we don't have the nucleotides to make it up. And something called primers, which you will know we have guessed because we've never come across that before. Right, so ATP is the easy one, we'll start with that, right? DNA replication requires energy, ATP provides that energy. The DNA template, okay, this thing here, the original, the starting molecule of DNA. You can't make a copy of something if you don't have the original to copy, right? So that's another easy one. We obviously have to have our original DNA, or sometimes it's called the template. We need to have 4 types of free DNA nucleotides, and they are required obviously to pair up with their complementary nucleotides. So if we look here on the picture, these here are our free nucleotides. So here we've got our original strand, or our template strands. Here we can see they've started to separate out, and here we've got our free nucleotides that haven't got anything to do just now, but they're quite happily, you can see that one there started to slot into place, and that one there slotted into place. Now obviously we need 4 types, as it says up here, and the reason we need 4 types is if there's an A there, then it's going to want to be a T that joins on. So if we have an adenine base there, it will want to be a thymine base there that links in with that. If that's a cytosine, it will want to be a guanine. And that's the reason we have to have our G's and our T's and our C's and our A's, because we have to have all the options to pair up with whatever base is on the template strand. We need enzymes. Now, there are There are lots of enzymes involved in DNA replication, but thankfully, you only need to know about 2 of them. So we've got one called DNA polymerase. Now, the word poly means many. Okay, if you do chemistry, you'll come across that quite a lot. A polymer is a substance made from lots of repeated units joined together. And you know that A's is the ending for most enzyme names. So DNA polymerase, something to do with DNA, lots of things getting joined together, and it's an enzyme. Now, this bit at the moment will seem really confusing. So what does DNA polymerase actually do? It adds nucleotides to the three prime end of the growing strand. A primer must be present for it to start to work. Now, that's going to make absolutely no sense to you just now. We'll come back to it when we get a bit further on. The other enzyme you need to know about is something called ligase. And ligase is a bit like glue. It joins bits of DNA together, it sticks bits of DNA together. And you might have heard of it before. I'll give you a little second to think. You may have heard of it before in National 5 genetic engineering, because ligase is the enzyme that's used to glue, join the DNA from one organism into another organism's DNA. You didn't need to know it specifically for National 5, but your teacher might have told you. And then we've got this new word, primers. Okay, now to prime something, you may already know, means to make something ready for use or ready for action. So decorators might prime a wall before they paint it, make it ready, fill in all the holes and any funny marks there are, make sure it's all kind of a nice blank canvas. Or you might get primed before you go for an interview, somebody might prepare you. So the primers are what we need at the start. Okay, we need them at the very beginning for DNA replication to happen. Primers are actually RNA nucleotides joined together. Now, you haven't really got a clue what RNA is yet, and it's not really that important for DNA replication, but you will see it described as that. So primers aren't DNA, but they're very, very similar. And just like the word to prime, to make something ready, we need these primers to get the DNA ready to be replicated. And there's another really important bit here. Primers bind to the 3' end of the template strand. Now remember when we did the last topic, we said that the 3' end and the 5' end and the fact that the DNA was anti-parallel was going to become really important in the next topic. So this is that moment. Right, so here we've got a really simplified diagram of DNA. So if we say this is our 3' end here and we follow that up, then we're going to go to 5, obviously. And if this is 5 down here, then hopefully when we follow it, we go to 3. Okay. So this picture is obviously not showing all the nucleotides, it's just showing a DNA as a line, but that's okay. So what we can see here is this arrow here is saying the DNA is unwound and unzipped. So rather than it still all being as a double helix, it's untwisted or unwound and it started to unzip. And when it does that, it's obviously easy for us to see that this is 3' end and this is a 5' end. Okay, and we've got here this word discontinuous and continuous. So you're thinking, well, why is there a discontinuous and a continuous strand? And then you're thinking, what did she just say about 3' and 5'? What did that thing say about the primers? Primers bind to the 3' end of the template strand. So here, primers are only going to want to bind to that side there, that 3' end. They're not going to want to bind to this 5' end because that's not what they do. They're going to want to bind further up, which is why we have this discontinuous strand and this continuous strand. Now, I know you're still really confused. That's okay. Okay, here we've got the diagram of DNA showing the nucleotides and as if it's already unwound. right? So it's not showing it as a helix anymore, it's unwound, but it's still zipped together, right? There's still all these bonds here, which I'm hoping you're thinking in your head, oh yeah, remember they're the hydrogen bonds. 5', 3', 3', 5'. This is the diagram that I told you, keep practicing, sketch it out, scribble it out, just whenever you've got a spare 30 seconds and a scrap of paper, just have a wee scribble of it. Include the 5 and the 3 and the 3 and the 5, and it'll just become second nature to you. Okay, so these primers are needed to start DNA replication, but they will only bind to the 3' end. 3' end, 3' end. Okay, so a primer is only going to want to bind here and then start DNA off in that direction, DNA replication off in that direction. Right, so how does it work? Okay, I'll just let you have a read of that. So sometimes the diagrams might say continuous and discontinuous, but this is where we get these terms leading strand and lagging strand. Right, this is where we get into the nitty-gritty. Now, every time we do a complicated process like this, you will get the words to describe it, and you'll get a diagram that shows it. And it's entirely up to you what one you choose to pay more attention to. I'm definitely a diagram type of person, so I would keep drawing out that diagram, but some people like the words and the diagram confuses them. So when you get your notes, you'll have both, okay? And you might want to sort of add wee bits in and whatnot. So let's go through this. Right, so we're going to start with the leading strand. So if we just go back, okay, so we're going to start thinking about this side. What is going on? This blue line here, what's actually happening? Where does that come from? How does it work? Right, so already said that when DNA wants to replicate, it has to unwind or untwist, and it has to unzip, okay, as the hydrogen bonds break between the bases. That's going to form two template strands, two strands that can be copied. This process occurs at several locations on the DNA molecule, and we've already said that. We looked at the diagrams of that. A primer will bind to the 3' end of the template strand, and a primer is a short sequence of RNA nucleotides, and we're not going to worry ourselves too much about that. So here we have, this is the 3' end of our template strand. We're always going to get in the habit of drawing it, so we have 5 at the top, at the left-hand side, 3 at the bottom. Here, this red thing, oops, is our primer. This little red thing here is our primer. And it wants to bind to the 3' end of the template strand. But once it's bound, we would say that's now the 5' end, because that's going to be a complementary strand. So that's the 5' end of the primer. This is the 3' end of the primer. And these little things here are the free DNA nucleotides. Now, you might want to pause the video just now and watch link 1. I've learned my lesson that these don't work if I put them into the video. So in the file section, I'll post the links, right? So you might want to watch that just now. Okay, the next thing that happens is our enzyme. The first enzyme that we learned about comes in. DNA polymerase starts to walk along the template strand, adding new complementary nucleotides to the template strand in the 5' to 3' direction of the new growing strand. Now, this is where it gets a bit confusing. If you're going to mention the direction, 5' to 3' or 3' to 5', it's really good practice to mention are you talking about the template strand, or are you talking about the growing strand? Because they're obviously going to be different. So if we just look at our labels again. Here we've got, so this side is our template strand. That side there is also a template strand, but we're not worrying about that side just now. We're doing this side. That's our primer that we saw joined on at the start. And this green thing here is our DNA polymerase. So remember, the first thing to happen was the primer joined on at the 3' end, this end of our template strand. The DNA polymerase, that's what this blurb up here is saying, then starts to walk along. So the DNA polymerase is then moved along here and added complementary nucleotides as it goes. So if there was an A on this, it will add a T on this side. If it was a C on this side, it will add a G on this side, and so on. And then this diagram is just showing it a little moment later, and look how far up the DNA polymerase has got. Okay, it's just continued to walk up, adding more free nucleotides. Now, you might notice that what was red here, the primer was red here, is now turned to purple. And that's because eventually the primer gets replaced by DNA, because remember we said the primer was actually made of RNA. Don't worry yourselves too much about that. The only reason I'm mentioning it is because it is on a lot of diagrams that you look at, and I don't want you to get a bit stressed out about and not understanding why. Okay, and then the last thing that would happen to that strand there is it would twist back up to form its double helix. Okay, so that strand there is now done. Okay, or would eventually be done if we continued on with the diagrams. Now that's quite straightforward. That's why it's called the leading strand. Oops. Now, remember, replication on linear chromosomes happens at several points along the strand of DNA. So what this diagram is trying to show is, here we've got our original molecule, 2 strands of our original molecule in blue. And here it's showing, that's the blue strand, and that's one. And so it's just kind of zoomed in a bit. And these little, lines here are representing the hydrogen bonds. And you can see that replication is happening here and here and here and here. Don't worry about these terms, origin of replication and replication bubble and replication fork. I've deliberately left them on there because when you watch videos and you maybe look at other images, they're all slightly different and they all mention different parts. You don't need to state any of these terms, but if you're familiar with them, it might make your life a little bit easier, especially if you're using Scholar, they might mention them. Just be sure when you're revising to know all the terms that are in your printed notes. You don't need to try and remember all the extra stuff. The black lines here, there's one there and one's there. I think they're actually meant to be red with black outlines, but they look pretty dark. That's the new strand there, and that's a new strand there forming. This is just showing it a little moment later. You can see that it's continued, and you can see that it would eventually meet up and the whole chromosome would be replicated. So here we've got the blue original, or parental DNA they're calling it here, and a red new strand, and then here a red new strand and a blue original. Right, now the lagging strand. So if we go to this diagram here, so this was our strand that we looked at before. This was our leading strand. This is the one that was nice and easy. The primer wanted to bind here and it just went all the way up. Easy peasy. But when we look at this side of the DNA molecule, we know that it's running in the opposite direction to this side. So this was 5 to 3. This one's 3 to 5. So we know that the primer is not going to be happy binding there. So we can't just go up there like we did the last time. What has to happen is we have to wait for a little bit of the DNA to separate. And then the primer, if we look at this diagram, this one here, the primer will then bind and the DNA polymerase will add free nucleotides in this direction. So if we can imagine that this diagram here is showing what's happened after a little while. Okay, so it started unzipping, started coming apart, and this one grew, and then this one grew, and then this strand grew. DNA polymerase is still doing the same job. The primer is still doing the same job. It's just that the strand is having to grow in the opposite direction to this side. We've got an extra enzyme in this diagram here. It's yellow on the screen here, and it's ligase. I know the label is absolutely tiny. Okay, it says ligase there. Now, if we remember, we said that ligase was an enzyme that glued DNA together. So because this has had to be made in little sections, so as this has unwound and unzipped, a little bit's been made, then another little bit's been made, then another little bit's been made. You end up with these little fragments of DNA, and they need to get stuck together, and that's what the ligase does, it sticks the fragments together. And just like before, the primer, which was red initially, will eventually turn purple just to indicate it does get replaced with DNA. Right, at this point you might want to watch link 2. It's much easier, I think, to visualize DNA replication when you can actually see it in an animation rather than me muddling my way through lots of diagrams. Again, if I had my model, I would be fine. If we just go back to the description for the wordy people. So since DNA polymerase can only add nucleotides to the free 3' end of the growing strand, one of the new DNA strands needs to be formed in short fragments. So this description is very similar to the leading strand description. Well, let's just have a look at what's actually different. Okay. So the first bit, exactly the same. This bit's different. Many primers need to bind at various points. That means we get fragments produced. And the fragments need to be joined together by ligase. Right, so they're the things that are different for a lagging strand compared to a leading strand. There's just a bad biology joke. I'll just let you chuckle to yourselves for a few seconds. Okay. Here's another link number 3. This is a video by the Amoeba sisters. They're usually quite good videos. They obviously like to animate them in a humorous way. So we've got ligase, we've got primer, we've got DNA polymerase. What this video also talks about is something called helicase. You do not need to know about that. It's very difficult for me to find you a video that says exactly what you need to know, not too much, not too little. This one's quite good, but it does mention helicase. It also mentions the origin, primase, SSB proteins, topoisomerase and Okazaki fragments. You don't need to know any of those, so just ignore them and just try and get a sort of general idea of what's happening in DNA replication. If you wanted to learn them, feel free, but you do not need to know them for hire. That's just another diagram, just because I like to show you different versions. Here we've got the leading strand and the lagging strand. You can see the leading strand. Here's our primer, which bound to the 3' end of the original or the template strand, and it's just got one continuous line, and that's our DNA polymerase happily adding nucleotides to match up with a complementary base, and that will just continue to happen. Lovely. This side, we had to wait for it to unwind a little bit, then we got a primer slotted in, and the DNA polymerase does its business. Unwinds a bit more, primer slots in, DNA polymerase does its business. And obviously, we will need our extra enzyme, our ligase, to join the fragments together. Here's another joke, full of jokes today. chuckle, chuckle, chuckle. Okay, we're nearly there, we're nearly there. Right, so the sort of second big part of this unit is something called the polymerase chain reaction. Now it's just shortened to PCR, right? Just like we have DNA. Is there a nice easy short way of saying deoxyribonucleic acid? We have PCR, polymerase chain reaction. And this is an in vitro technique used to create many copies of a target region of DNA. So it's like a version of DNA replication, but it's in vitro. And you're thinking, well, am I supposed to know what in vitro means? Perhaps you do. You might have heard of in vitro fertilization, or IVF, which you may or may not know is when the fertilization, so the sperm and the egg, are made to fuse, or nuclei are made to fuse in a laboratory outside the body, in a test tube, in a petri dish. So in vitro means made to occur in a laboratory vessel or other controlled experimental environment, rather than within a living organism or natural setting. Okay, so this is the way that scientists can get a bit of DNA from somewhere and make it replicate outside the human body, so in a lab. And usually there's just a target bit that they're wanting to replicate over and over again. Right, so why would we want to do this? So there's some clues. I'm not sure you'll be able to figure out what that is. If you do, that's using an electron microscope, so it's an incredibly powerful microscope. Okay, how do we think of that? So PCR, what can it be used for? So it can be used, like we said, to amplify DNA, to make lots and lots and lots and lots of copies of DNA outside the body in a laboratory. So where would you get that DNA from then? Well, perhaps it will be from the scene of a crime. So if you're like me and you like your silent witness or your CSI, who done it and how do they figure it out, then you might know all about that. Embryonic cells, that's what that is, an embryo, to try and work out, to diagnose perhaps if the embryo has got a genetic disease before it obviously grows into a baby, and to settle paternity tests. Who's the daddy? So if you'd ever watched Jerry McKyle when it used to be on, we're constantly doing those tests. And quite obviously very much in our heads just now is the coronavirus, COVID-19. And if you go onto any site and sort of look at, how are the testing for it, what are these tests all about, then it actually mentions PCR in that. So this is just an image from a BBC site, BBC News site, and it's basically saying, what are the differences between the test that you get for COVID-19 that says you've currently got it, about the test that would test whether you've had it in the past and you're now better. Now, this have I got it test is the one that NHS workers, key workers can go and get now. This is the one they've had this target about, we want however many thousand tests per day. And you can see they take a swab from your nose or your throat. And what do they do with that swab? Well, they put it through a PCR machine. They're looking for viral DNA. Okay, it looks for coronavirus genetic material. So it looks for the DNA of the virus. And that obviously, if you've got the virus DNA in your nose or your throat, then you've got the virus. So I just thought that was quite interesting for you. Maybe not. Maybe I'm completely boring you. Right. So what do you need for PCR? Well, it's very similar to DNA replication, but it's not inside cells. Okay. So very similar. So have a little think of what you'll need. What do you need for normal DNA replication when it's inside a cell? Just so that you're aware. We've said that this is called, this is called in vitro. If something is happening inside the cells, then it's called in vivo or in vivo. So VIVO. That's just a random fact for you. Okay, so what do we need? You've had time to think about this. Okay, we still need the DNA template, and that's obviously going to come from the crime scene, or the virus is up your nose, or the embryo, wherever it is you wanted to test the DNA from. We need primers, we need free DNA nucleotides, and we need DNA polymerase. Right, so what's not in this list is ligase and ATP. Okay, we don't need ATP because it's not happening inside cells, and we don't need ligase because of the method used, the PCR method used. We're not going to have a leading strand and a lagging strand, so we don't need ligase to glue them together. Right, so what do they do? There's a lot of temperatures involved. So they put all their ingredients, right, all these things we've just said, they dump them all in to a tiny little tube, and they stick it in a machine. And that machine then heats the stuff up to between about 94 and 96 degrees Celsius. And what that does is it separates the strands. And you're then thinking, well, what kind of bonds are going to be broken when the DNA separates? And you would be right in thinking it is going to be the weak hydrogen bonds. So this tiny little diagram here, there's our DNA in blue, and there the two strands have separated out. So the hydrogen bonds are broken when it got heated up. Next, the temperature is reduced slightly. So we've gone down to 50 to 65 degrees Celsius, so that the primers can bind. So here, the green things here are our primers. So there we've got primer bound at that side, primer bound at that side. And that was, remember, with the temperature reduced a little bit. We then heat it up again, not as high as it was to start with. So we started really hot, then we dropped down, and then we've gone back up a little bit. And that then allows this red thing, which is called heat-tolerant DNA polymerase, can then do what DNA polymerase does, which is adds the nucleotides to the DNA strand. So you can see it's added these yellow bits on, the free nucleotides on to either side. Now, why does it need to be heat tolerant? You're asking yourself. You're thinking, wait a minute, what do I know about enzymes? Remember DNA polymerase, it's an enzyme. What do I know about enzymes? 72 degrees Celsius, does that look like it'll be good for an enzyme? Certainly not. So a normal enzyme would be denatured by that high temperature. So that's why we need to use a special enzyme called heat-tolerant DNA polymerase. Okay, so after one shot, you're going to have two identical molecules of DNA produced. You started off with one, that's going to replicate, you're going to have two. After the second cycle, each of those is going to replicate, and you're going to have 4. Then you're going to have 8, then you're going to have 16, and so on and so on and so on. And it very, very quickly, this little magic machine can make you lots and lots and lots of copies of that target DNA. And then you'll be able to say, yes, you have coronavirus, or yes, you are the daddy, or yes, they are the murderer, or whatever. This is just a different diagram. This is a wee machine, this is a wee tube. There's our DNA, that's separated, there's the primers bound, and there's our heat-tolerant DNA polymerase adding nucleotides in that direction, adding nucleotides in that direction. Now, because the whole thing's been separated, that's why the primers can bind at the end. They want to bind that, so we don't have this need for the ligase. Right, we've got link 4 and link 5. So link 4 is a good animation that looks at PCR, and link 5 is a song, because biologists love a song. Okay, so that's been hard going, guys. It's been hard going for me. It'll be really hard going for you, and I do not expect you to understand all of that. Okay, it's our starting point. So we've looked at what do you need for DNA replication. We've looked at how does it work on a leading strand? How does it work on a lagging strand? What are some uses of PCR? Why is it required? and how does PCR happen? How does the process happen? Okay. Right. Good luck.